Hi everybody, this is Mandy here, and we are on our fourth part of our Food Safety Info Friday, and we're going to be talking about the AOAC methods that are used to analyze your Listeria. So first, what is AOAC? AOAC is the Association of Official Analytical Collaboration International. The purpose of this program is that they have an official methods program, which is used to make sure that methods are credible, and all the methods that have an AOAC certification undergo review by a board of scientists. So I have a question for you guys. Do these numbers mean anything to you? They should, because these are the AOAC certification numbers that are used for the methods that analyze your environmental Listeria swabs. So Summit does 2 of these methods, and we do another one. But the important thing is that these methods were all created by a third party. Each lab gets to pick which ones they follow, but all these methods underwent AOAC certification. On the base level, no method is better than another. Each method has its own downfalls, but they can be used in different situations, which we will go over later. So let's start with this one. AOAC RI 051802. What is this? This is Solus 1 Listeria. This is made by PerkinElmer. And so here are a couple quick facts about it before we dive deeper into it. First, it has an incubation period of 22 to 30 hours. Sample heat and cool time is about 2 minutes. An assay procedure and preparation take at least 2.5 hours. And the final analysis time is about 2 hours. And this is the kit over here that PerkinElmer will provide you once you order it. So let's get into how Solus 1 actually works. First, there is an enrichment step. So first you add in Solo Plus, which is just their special magical mixture, to a sponge, which the sponge is the one that you swabbed with and had your sample on it, and you incubate that for 22 to 30 hours. Next, you're going to transfer 1 milliliter of the solution into a boiling tube for 15 to 20 minutes, and this basically, denatures any proteins. It destroys those Listeria, so it's no longer living. And then finally, the sample is then cooled to room temperature. All right, for this method, we are going to throw back to Biology 101 a bit so that we can get a better understanding of how this method actually works. So this method is based on antibodies. So antibodies are everywhere. We've been hearing a lot about antibodies since COVID-19 came out. In the basic sense, an antibody is a protein that is produced by cells after the stimulation by an antigen. So what does this mean? Here you have your antibody and you have your antigen. Now we can see that this antigen fits perfectly at the end of this antibody. Here we have a bunch of other antigens, and we can see that this red one won't fit in there, a green one won't fit in there. So this is important because every antibody has a matching antigen. So vaccines, for example, you get a vaccine, it gives you one of these matching pieces, and then your body creates the other matching piece to it. So this is very important for this Solus 1 Listeria. This is the basis of how this method works. Okay, so now we are into our immunoassay portion of Solus 1 Listeria, and our first step is we are going to prepare our wash buffer. So this wash buffer is a mixture that basically cleans up the sample. It removes any debris or anything else that could interfere with analysis. We are going to add in our blank, negative, and positive control, and these just ensure that the test worked correctly. There's no sample in these controls. Step 3. We are going to transfer the sample and incubate at 37 degrees Celsius for 60 to 65 minutes. So we are going to take our sample out of our boiling tube and put it into each of these wells. And each of these little dots are single wells in the microplate. Next, we are going to remove this liquid and we're going to wash the cells 5 times. Washing is done with a wash buffer, so this is removing any extra sample or debris or anything that could interfere with our analysis. We're going to add in the conjugate and then we're going to incubate this plate at 37 degrees Celsius for 30 to 35 minutes. So when I said removing extra sample, you're probably wondering why would we remove sample? Well, this is why. This is what is happening in our wells on our microplate. So this is an individual well. At the bottom of each well, we have bound Listeria antibodies. So what we do is when we add in our sample, we are also washing away any extra samples. So what do I mean by this? When we add our sample, the sample has antigens in it, and these little purple dots are Listeria antigens that came from the ruptured Listeria cell. They will bind specifically to those bound antibodies to the well. When we wash, we get rid of any extra Listeria antigens that are present because we don't want those interfering with the test. We only want the antigens that are bound to the Listeria antibodies that are bound on the well. Now we're going to wash again, remove anything else, and we are going to add in our conjugate. So what is our conjugate? Our conjugate is this black structure right here. So we have an antibody with a bound enzyme. E stands for enzyme. This antibody will only bind to the Listeria antigen that is bound to the Listeria antibody that is bound to the well. So we have our conjugate bound to our antigen, and our antigen is bound to the Listeria specific antibody that was bound to the bottom of the well. Next, we're going to wash the plate again, and we're going to add in our substrate. So over here, we have our complex here with our conjugate bound to our antigen and our antibody. We're going to add in our substrate, and remember, an enzyme is like a factory. It turns one product into a different product. So it makes apples into apple pie. So we start with our substrate, and it will get turned into a different product. This reaction only occurs if this conjugate is bound to the antigen-antibody complex. If it's not bound, this reaction will not occur. Finally, we have our last step, step 6, where we incubate the plate in the dark for 30 to 35 minutes, and we add a stop solution. What this stop solution will do is it will turn this previously blue-colored mixture into a yellow color, and this is done because the newly made product from the substrate, from the original substrate, will turn yellow when a certain chemical is added to it. So this test uses the Dynex DS2, which is the machine which determines the optical density of each well. Optical density is the measure of absorbance. So here's how this works. We have our light source, our lens, prism, and wavelength selector, our sample, and our detector. What will happen when we turn the machine on? The machine will emit a light source. It will pass through the lens, which will be directed into the prism. The prism will then separate that beam of light into its individual wavelengths. Our wavelength selector will select for the specific wavelength that we want. In this case, it's 450 nanometers. So only light that has a wavelength of 450 nanometers will pass through the selector, through the sample, and then the detector will determine how much the sample is absorbing that light, and then we get a reading. So basically, this test will tell you if a sample absorbs more light, then there's a greater amount of Listeria. Because remember, we had our sample turn from blue to yellow, and the change in yellow was based on if the conjugate was bound to the Listeria antigen antibody, which allowed for the added substrate to be changed into a different product. That product turned a different color when we added our stop chemical. So again, the greater the color change, the greater the amount of Listeria antigen present. So for SOLAS 1, we have our optical density reading of 450 nanometers. So if our optical density is less than 0.2, it's a negative result. If our optical density or absorbance is greater than 0.2, it's a positive result. And again, we have our control readings, which we need a working control to be sure that this test worked correctly. This is our AOAC certification for SOLAS 1 Listeria. So we have our certification number and a bunch of other details that were part of the report. All right, that was just one method. Now we have our second method, the AOAC 2016.07, and this was made by 3M. It's a molecular detection assay to Listeria species. So it's made by 3M, and our incubation time is 24 to 30 hours, our sample heating time about 22 hours, and our assay time about 75 minutes. So this is kind of the whole setup or kit that you need to do this. Okay, so our first step, we're going to start with our enrichment. So we're going to add in the enrichment media, and we're going to incubate that for 24 to 30 hours at 37 degrees Celsius, and this is simply to increase the amount of Listeria species to a detectable level. Next, we move into our lysis phase. So hopefully everyone's super familiar with lysis by now. So we are, again, bursting open the cell and releasing all the intracellular compounds, and the release of the DNA from the cell is important for the 3M method. So we're transferring 20 microliters of the enrichment media into the lysis tube, placing those lysis tubes at 100 degrees Celsius for 15 minutes. Also, we have a negative control, and this negative control is simply just sterile enrichment media, so it should give you a negative result, and this just makes another checkpoint to make sure that the test is working properly. When you place the tubes in this heat block, they start off pink and they will turn yellow, and yellow means that the samples are heating up. After 15 minutes, we're going to remove them and cool them down to 20 to 25 degrees Celsius for 5 to 10 minutes. And the color will change from yellow back to pink. And then finally, we're going to remove these tubes from the cooling. Next, we have our isothermal amplification. So this means that during amplification, it stays a constant temperature during this amplification process. So we start with our reaction tubes at room temperature. These reaction tubes are super similar to our Listeria right now tubes, which are pictured over here. The pellet, again, has all the compounds needed to perform this assay. So we are going to transfer 20 microliters of our sample from our lysis tube into our reaction tube. We're going to cap it, place it in the speed loader tray, and then place it in the 3M molecular detection system pictured down here. What will happen in the machine is a process called loop-mediated reaction, and this is for target DNA in the Listeria. So the amount of DNA is going to be measured by bioluminescence. So instead of me trying to explain this procedure, this LAMP procedure, I'm just going to show you a video because it explains it fast, quickly, and as simply as it can be.
When designing a LAMP experiment, 4 to 6 primers are required, specific to identified regions of target DNA or RNA sequence. Amplification amplification initiates from strand invasion by one of the inner primers, and a strand displacing DNA polymerase extends the primer and separates the target DNA duplex. The first product is then displaced by synthesis, initiating from an outer primer which anneals to an upstream target region. As it is displaced, the end of the product forms a self-hybridizing loop structure due to inclusion of a reverse complementary sequence in the inner primer sequence. This annealing and displacement cycle repeats on the opposite end of the target sequence, and the resulting product is a short dumbbell structure that forms the seed for exponential lamp amplification. This lamp dumbbell structure contains multiple sites for initiation of synthesis, from the 3' ends of the open loops and annealing sites for both the inner and loop primers. As amplification proceeds from these multiple sites, the products grow and form long concatenamers, each with more sites for initiation. The result is a rapid accumulation of double-stranded DNA and amplification byproducts that can be detected by a variety of methods.
So I hope that made a little bit of sense. I know there was some crazy lines going all over the place, but the base message I wanted you to get out of that was that you start with a low amount of DNA and that gets amplified into a great amount of DNA. It's not showing the video, but then a probe will come in, bind to the DNA, and that once that probe is bound, it will fluoresce. And that bioluminescence is the basis for how this test is measured. Listeria right now uses a similar method to this. They also use bioluminescence, but the only difference is that this method uses lamp amplification, whereas Listeria right now uses a slightly different method of amplification. So here we have our AOAC certification. So we have our performance test method and our official method of analysis certification. So now we have Listeria right now, which is AOAC 081802. This is made by Neogen, and this is what we do here at Riveridge. So we have a sample heat time of about 42 minutes and an assay time of 18 minutes. And we can see here the kit that I use in the lab. So since we already covered Listeria right now in the last Food Safety Info Friday, I encourage you, if you do not remember most of it, to please go back and review it, because we're just going to fly through it real quick since we already covered it. So first step, we're lysing the cell. Remember, we're breaking open that cell membrane and releasing all the intracellular components into the environment. We're transferring 500 microliters of the lysate, the sample components, into the lysis tubes. We're heating those at 37 degrees Celsius for 10 minutes. Next, we're transferring those tubes into a 80 degrees Celsius heat block for 28 minutes. And finally, we are going to heat reaction tubes that do not have the sample in them at 56 degrees for 4 minutes, just to get those enzymes working at their optimum temperature. We're next going to transfer 50 microliters of lysate to these reaction tubes. We're placing the reaction tubes in the answer machine, and we let the machine do the rest. During this, we have our isothermal amplification, meaning that it stays a constant temperature throughout amplification. We have our rRNA that is reverted to its complementary cDNA. So I encourage you, again, if this you don't quite remember this, to go back and look at our previous video about Listeria right now. And here, remember, this is measured by bioluminescence. So we have our DNA fragments that were made from our rRNA, and we have our probes that were in our reaction tubes. And our probes, once they attach the DNA, they fluoresce, and that fluorescence is measured by the answer machine. Here we have our Listeria right now AOAC certification. So again, the same certification as the Solus 1 Listeria and the same certification as the 3M molecular detection assay. So we have our certificate number, and we have just some other information over here about the certification. So that was a brief overview of the 3 different methods that we use to analyze your Listeria. Again, all of them are AOAC certified, and in one of our upcoming food safety Fridays, we'll dive deeper into what it means to be AOAC certified. We also want to get into, okay, well, what test do I use now? There's 3 different methods and they all seem pretty different from each other, so what do I do? When do I use it? So we'll get into that next time in our upcoming series. But to give you a quick answer, the main question, how fast do you want to know? SOLAS 1 is going to give you a next day to 2 days from collection. This is depending on when the lab gets the sample. The 3M molecular detection assay, again, it's going to be either next day or 2 days from when you collected the sample. Listeria right now is going to be the same day as you collect the sample. So this one is an immediate result. So again, in the future, we're going to go over maybe when to use SOLAS 1, when should I use the 3M method, when should I use Listeria right now. And we're going to go over a case study that can kind of help answer some of those questions. Thank you for watching. I hope this helped you and you guys learned a little bit more about how people are handling your environmental swabs when you give them to the lab. For some upcoming food safety info Fridays, we are going to discuss how do I pick which method I want the lab to use, the AOAC method accreditation process. All these methods go through the same process, so no method is better than another. They each have their downfalls, but we're going to dive more into that in this episode. We're going to get to steps to improve your environmental program, creating a site list and just, really emphasizing that, this stuff does matter because it affects your everyday processing. And we're finally going to do our ATP testing and our trending. So stay tuned. Thank you for watching. Please reach out to me, Jamie, or Teresa with any questions, or if you want more information, we would love to get that together for you. Thank you.